Hello, can I have some questions? Hello, are there any people, the students here? Hello, sir. Are there any students here? Hello, sir. So, good morning, one and all. So, this is the syllabus of sericulture, an introduction to silkworm breeding. So, today I'm Dr. Vanita. I'll be taking up this particular topic. So, I'll be dealing about silkworm breeding, and in this topic, we are having major aspects of silkworm. So, first of all, about silkworm. So, I'll be sharing the PowerPoint presentation. So, this is information about silkworm breeding. So, in silkworm breeding, as we know, silkworms were first domesticated in China over 5,000 years ago. Since then, the silkworm production capacity of the species has increased nearly tenfold through artificial selection and intensive breeding process. So, as you know, the scientific name of silkworm is Bombyx mori. And when we happen to see the life cycle of silkworm, we have the silkworm. It gives around 350 eggs, and the eggs larva comes out of the eggs after a period of time. And this larva undergoes a phase where it is rapidly eating the leaves, and it undergoes a phase called molting, and after which it forms a caterpillar stage. This caterpillar stage is a profusely eating worm, and then it forms a cocoon stage. It is this cocoon which produces the silkworm. So, after knowing about this, we have eggs. An egg is the first, as we know, the information about the life cycle of the silkworm. So, our primary interest is the silkworm. So, our primary interest is the silkworm, which we get in the cocoon stage, right? So, this cocoon stage is a primary interest because in this stage there are the silk which is wound, and uh, it is said that around 800 meters to 1200 meters of silk is there in the cocoon. So, so for the silk, it is put in hot water so that the worm dies and the silk is easily, readily taken and sent to the reeling yards. So this is how the silk is extracted from the silkworm. So what happens if silk is not extracted from the silkworm? If silk is not extracted from the silkworm, it produces the as a cocoon forms at an adult, it the wires break up. As a result as a result all the silk is broken into small pieces. So in China these are also used in some kind of cushions. Anyhow, for a good quality of silk, we require the silk which is not torn. For this reason, it is put in the hot water. Silkworm breeding. As we see the history of silkworm breeding, the goal of silkworm breeding is to maintain maximum productivity in the yield and quality. Though silkworm is introduced its commercial venture way back in 17th century, silkworm breeding was initiated in 1920. This goal is achieved by bringing genetic improvement through combination of desired genes by crossing two selected pure stocks of the silkworm followed by selection. So as you see, we require a good quality of silk. For this reason, there is constant improvement genetically by combining the desired genes, which is achieved by crossing the two selected pure stocks of silkworm. As we are aware that in this life cycle of silkworm also there are a number of health hazards to the larvae, the different stages in the life cycle. So, if all the stages in the life cycle, these pupa, egg, all are correctly they reach the pupa stage, we get a very good quality of silk. But there are sometimes there are possibility that there are deaths of larvae due to some kind of infection. So this due to this infection, there might be decrease in the yield. So, this silkworm is an organism where the principles of genetics and breeding are applied in order to harvest minimum output. It uses the principles of heterosis and crossbreeding. So, traditionally, silkworm breeding has been focused on achieving high production with a better quality while improving survival, reproduction, and physiological characteristics. So, as you are aware about polyvoltine and multivoltine, bivoltine, so there are some of the polyvoltaic breeds which are characteristics of India. So these are pure Mysore, Nichi, which are very popular breeds of South India. Apart from that, we have Nistari breed, which are also very popular in West Bengal. In addition, Sarupat and Mornia in the northeast were rare through these breeds were well adapted in both respective regions. So now we'll be coming to know about inbreeding. Inbreeding is a concept where mating of individuals or organisms are closely related through common ancestry. Now, inbreeding is useful for the retention of some of the desired characteristics or elimination of undesirable characteristics. But it can also lead to decreased vigor, as we know that inbreeding it brings more of homozygous characteristics in the offspring, so it could lead to some kind of depression, genetic depression, and it could lead to, besides advantages in the disadvantages, it could lead to some kind of mutations in the organisms which could help to bring out the recessive characteristics. Both inbreeding and hybridization are forms of non-random mating and selective mating, but they operate in an opposite way. Inbreeding is a kind of genetic assortic mating, which is compared to the phenotype assortic mating of hybridization. While gene frequencies do not change on the whole, genotypic frequencies do change towards the production of more homozygotes and fewer heterozygotes. As you know, as it is the worms are being mated within the same ancestry, we find more of heterozygotes and fewer of heterozygotes, more of homozygotes and less amount of heterozygotes. Thus, any change in the population mean as a result of inbreeding must be related to the difference in the genotypic value between the homozygote and the heterozygote. Outbreeding refers to mating between individuals from different populations, subspecies, and species. Now, what are the uses of outbreeding? As we have seen, inbreeding is a type of breeding where the focus is purely to bring out some kind of desirable characteristics. As a result, mating is there between silkworms within a common ancestry. In contrast to inbreeding, we have outbreeding, where there are mating of silkworms of totally different species or within the species, but they are not related. So this outbreeding can result in reproductive fitness known as outbreeding depression. Could lead, but it can mostly lead to hybrid vigor and new amount of heterozygous zygotes could be seen in the population. So in this manner, we find many advantages of outbreeding. Now, when we compare inbreeding and outbreeding, inbreeding is the mating of more closely related individuals, and outbreeding is the mating of unrelated animals. Mating is mating is between animals of the same breed for few generations, whereas mating is for different organisms, different it could be species or different breeds. So in outbreeding, they are different as till 4 or 5 generations they are not from the common ancestor. Advantages is that it increases homozygosity, whereas advantages of outbreeding is that it leads to heterozygous characteristics. So, a disadvantage of inbreeding is that it could lead to inbreeding depression, and disadvantage of outbreeding is that it could lead to outbreeding depression. So, mating is seen between same species, mating is seen between different species. So, these are the different characteristics of inbreeding and outbreeding. So, outbreeding again is classified into many types. One of the type of outbreeding is outcrossing. So here the mating is between the animals of the same breed but not having common ancestor for 4 to 5 generations. It is usually used for animals which have below average productivity and growth rate. It helps to overcome inbreeding depression. So as you know, crossbreeding, it produces more of hybrid offsprings. So here the mating is between superior male of one breed and superior female of another breed. So this results in the formation of a hybrid which may have more of more of desirable characteristics of both the parents, and it is having a better productivity. As we see, the another type of outbreeding is interspecific hybridization. So here the males and females of different but related species are mated. The progeny has desirable features of both the species. And examples of these are mule is a cross between donkey and horse. So it has many characteristics, but so also we can also find that it is also sterile. So the main objective of silkworm breeding are improving the fecundity, improving the healthiness of larvae, improving the quantity of cocoon and silk production, improving the quality of cocoon and silk production for specific purposes based on cocoon and silk production for disease resistance. So, as when we mean fecundity, it refers to the egg laying capacity of a breed. It is very important factor since commercial sericulture is strongly dependent on silkworm egg. So, we mean to say that how commercially how the kind what kind of egg laying capacity silkworm that particular breed is having. So that is called fecundity. Healthiness of the larvae. Healthy larvae lead to healthy cocoon crop. So healthiness is dependent on factors such as better pupation rate, less number of dead larvae in the better pupation rate, less number of dead larvae in the mountage, shorter larval duration. So as you see, this shorter larval duration is important as it reduces the chances of infection. And quantity of cocoon and silk. So as the silk production is directly proportional to the quantity of cocoon. So, so this quantity of cocoons will be more when we have a healthy larva. Coming to the quality of cocoon and silk, it depends upon number of factors like including the genetic factors. Now, specific purposes. There are some specific purposes behind this kind of inbreeding or outbreeding. So, we can know some of the specific purposes. Apart from the commercial purposes, advanced countries are giving attention to specific breed development or specific purposes like the sericin production or the protein which is there in the silk and sex limited breeds, thin or thick filaments production. So we find that there are a number of commercial purposes where we could give importance to the protein of the silk production, or there are some kind of breeds which are where there are some kind of qualities which are sex limited. In addition, we also find thick and thin filament production. So some silkworms produce thin filaments, whereas some silkworms produce thick filaments. So this could be used uh, in inbreeding or outbreeding to produce that particular variety of silk. Disease resistant breeding. The major reason for crop loss is pathogen infection. Efforts are there to resolve breeds or to evolve the breeds which are tolerant or resistant to various pathogens. According to Leo 1993, there are at least five quantitative characters that are of interest to silkworm breeders. These are color of the egg, larval markings, blood colors, color of cocoon, cocoon size. So when we happen to see the color of the egg, so these are usually the combination of the shell color, which are semi-transparent to opaque or yellow color or sometimes green color. So, so these could be used for taking the characteristics behind the egg color. The second are the larval markings. So these are normally of P plus, these kind of alleles with various markings to plain without any markings. So the gene P is multi-allelic cis condition. Another important characteristic which can be taken is the blood color. As we know, insects have hemolymph and the blood color, there is no hemoglobin present. So there are two major independent genes of the blood color which are yellow and white. Now fourth is the cocoon color which are normally white, green and yellow. So these are also taken for consideration during inbreeding. And next one is the cocoon shape. So there is so much of variations in the cocoon shape depending upon the species. Most spherical cocoon are found in Chinese varieties, whereas peanut-shaped cocoons are found in the Japanese varieties. More peaked cocoons are found in the tropical varieties like India and in Southeast Asian countries. The cocoon shape inheritance is controlled by polygenic additive gene actions. There are also a wide range of variations in size and surface tension, with double cocoon semi-dominant over single. So we have seen that female cocoon is yellow and male is white. So it is easy for sex discrimination and the male cocoon can separately be reared to produce high grade silk. So, in order to identify the male and female, so this is very good characteristic where we find the female cocoon, which is yellow in color, and the male cocoon is white in color. So it is very easy to discriminate between the male and female cocoons. In the recent times, the effort of silkworm breeding at sericulture experiment station in Bridza, Bulgaria, were directed to create lines of hybrid having higher tolerance to adverse breeding conditions. So, as we see, Bulgaria, Bridza, uh, is a place where there is sericulture experiment stations where there is constantly some kind of experiments being done to improve the yields of silkworm. So as we see, in the northern parts of the world, the temperature is cool, whereas in the southern parts, the temperature is hotter. So these silkworms are adapted to the climatic conditions. So on breeding these different kinds of silkworms, we get a silkworm which is adaptable to the diverse climatic conditions. So, so there is a one instance, one example of inbreeding, which is there here, where in the initial population, a cross is taken between the female of Nick1, which is a sex limited for larval markings, and female marked, whereas the male is plain and the larva is plain, and these are the Japanese varieties. So having the male and female with two different breeds, NIG1 and KK male, we produce the FN generation. So after the FN generation is produced, it is again mated to to the parent, which is a male parent, and this process is repeated for 9 generations. For example, after we get the offspring of FN generation, it is again mated. The offspring is again mated to the male KK. Then their offspring is further mated with the male parent. In a similar manner, this kind of process is repeated till 9 generations in a batch rearing. That means in a large scale they are being reared. After this, there is a selective breeding where they are reared for a small population for three consecutive generations. That means after 9 generation we have got the F9 generation. So this generation, this offspring is again mated with the parent, male parent, further which their offspring is further mated to a male parent, and further which it is also mated with the male parent. And the this particular generation is a breed which is obtained, which is IVA1. So this is a breed which is called as a pure line species, and it is further used in further experiments and used for the further studies. Now we'll be knowing about what is line breeding and mutational breeding. So line breeding is a form of inbreeding that involves selection of mates on the basis of their relationship to a certain superior ancestor. The back cross, or crossing a first generation hybrid with one of the parental types, is a common method of inbreeding. So, as we know, backcross is the first generation hybrid. It is being crossed to the parental type. So, this backcross is the type of crossing which is being done in line breeding. In addition, there are some more kinds of crossing which can be done in line breeding. For example, the breeding between individuals in the same ancestors, or the breeding between descendants or the cousins. So this also is followed in line breeding. So this method can be used to increase a particular animal's contribution to a population. So, how could we differentiate line breeding to inbreeding? In inbreeding, we are having breeding where the animals are bred within the closed siblings or the closer ancestors which are quite closer in their genetic relation, whereas in line breeding there could be cousins or there could be back cross. So in some aspect line breeding has a more of heterozygous characteristic found in the offsprings. So, line breeding is less likely to cause problems in the first generation than the inbreeding. So, as we know, inbreeding can cause more of genetic disorders in the offspring, whereas line breeding, as we have seen, it is connecting the uh, connecting the breeds which are little distant compared to inbreeding. Over time, line breeding can reduce the genetic diversity of a population and cause problems related to a too small genetic pool and may include an increased prevalence of genetic disorders and inbreeding depression. So, over time, line breeding could also reduce the genetic diversity and could also increase the chances of genetic disorders and cause inbreeding depression. Now we'll be knowing what is mutational breeding. So, as the name indicates, mutational breeding is a breeding where preference is given to mutations in the offsprings. As mutations are the result of production of more number of hybrid species. So this concept is widely used in production of more and more new offspring having a better quality of yield, which could give a better resistant type of silkworms and a better quality of silk. So mutational breeding is widely used in sericulture to increase the yield as crossing between breeds. The purpose is to produce some kind of genetic mutation in order to yield a new offspring having all the desirable traits and characteristics. So, as we have seen, increase in efficiency of silkworm and is a basic product of sericulture industry, and it requires the employment of scientific methods of breeding, provision of standard uh, conditions for adequate nutrition and maintenance of environmental temperature, adequate humidity, light, sanitation. All these are required, and the use of genetics and breeding for accumulation of desirable genetic factors in commercial species of silkworm. These days, conservation of genetic resources is one of the important goals of breeding science. As we know, genetic resources are the basic aspects of breeding. So, a good quality of silkworm, which produce good silk and disease resistant, which are able to adapt to diverse climatic conditions, are the need of our. So, this is used as a basic requirement in the sericulture. So, we have seen about different aspects in today's class. So, we have seen about what is inbreeding and outbreeding. Then, we have seen the difference between inbreeding and outbreeding. So, inbreeding, as it refers, inbreeding is a breeding where we are using the breeds within the same ancestry to produce offspring having a pure line species. In addition, we have also seen outbreeding where there are uh, offsprings produced from different species. So basically, both inbreeding and outbreeding are meant for specific purposes of sericulture and silkworm breeding. And both inbreeding and outbreeding have their own advantages and disadvantages. So, as we see the types of outbreeding, we find there are three major types of outbreeding, which are outcrossing, crossbreeding, and interspecific breeding. So, in outbreeding, there is a mating between animals of the same breed but not having a common ancestor for 4 to 5 generations. It is usually used for animals which have below average productivity and growth rate. It helps to overcome inbreeding depression. So, as they have seen crossbreeding, it is a mating between superior males of one breed and the superior females of another breed. Superior qualities of both the breeds combined, and this is known as hybrid vigor. As we also see between, so this is in context of um, silkworm. So, if we happen to see a silkworm which is having a thick uh, silk and which is from China, and we are mating with the breed, Indian breed, which is a disease resistant breed. So we are crossing these two breeds, and relatively we are getting the offsprings having a better quality of silk and also which is a disease resistant. So either ways we are getting the hybrid which is having the superior characteristics of both the male and female. So this is called as cross breeding. If we quote another example of any kind of a cattle, so we find like Jersey cow gives a high yield of milk. So crossing between a Jersey cow and a bull, a very good bull, can give a desired high yielding milk variety in cattle. In a similar manner, we are also using this particular crossbreeding technique in mating between various aspects of silkworm. We also find the qualities related to uh, volatinism, that means number of breeding in a year, as well the quality of the silk. So we want uh, more number of silk in a year, which is called as polyvolatinism. And with this concept, we also want a silk which is of a very good variety, which is a thick silk. So we are taking the breed from other regions like regions of China, and we are crossing with the Indian breeds of silkworm. So the resultant breed is a breed which is having a very good silk, as well as it is having number of times it is breeding in a year. It is having the characteristics of polyvolting. So this is crossbreeding extremely useful in sericulture. So as we have seen here, the hybrid aspect being applicable or the examples of Sardale sheep, which is a hybrid of Bicaneri eves and Merino rams. To explain the concept of hybrid, this illustration is given. So interspecific hybridization, as we have seen, the males and females of different but related species are mated, and the progeny has desirable features of both the species. For example, a mule is a hybrid of donkey and a horse, and it is more stronger and more disease resistant than a horse. We also find a liger, which is a cross between a tiger and a lion. So this is an example of interspecific hybridization where The males and females of different but related species or unrelated species are mated. So the resultant is a hybrid which is having a better yield, but this hybrid can also have uh, genetic depression, or it the offsprings could also be sometimes sterile. So these are the three different types of hybridization techniques mentioned in the outbreeding techniques. Outcrossing, the second one is crossbreeding, and the third one is interspecies hybridization. When we move on to some of the important objectives of silkworm breeding, we find fecundity, improved healthiness of larvae, improving quantity of cocoon and silk production, and improving quality of cocoon and silk production. In addition, for specific purposes like cocoon and silk production for disease resistance. So, the purpose behind silkworm breeding has some of the important objectives where we are taking care of the fecundity, that means the number of eggs being laid by the silkworm, and so as the egg form the larvae, we are paying attention to the healthiness of the larvae. So here we want improved health of the larvae so that they are later mounted into the pupae stage. So at each step of the life cycle, utmost care is taken so that there is rapid transition from egg, larvae to cocoon, and then we get the desired silk. So this is the importance of taking care about the objectives or the core areas of silkworm breeding, where we have seen about the fecundity of the egg or the importance of the eggs, and then we have seen about the larvae, how healthy the larvae is. Because if the larvae dies, we will not be able to achieve the desired silk. We will not be able to form, the larvae will not form to cocoon and will not be able to get the desired silk. The quantity of cocoon and silk production, in order to get a good quantity of cocoon and silk production, we require the larvae to transit into the cocoon. For this reason, there are utmost care being taken so that there are no kind of any kind of infection in that particular area. So these are the primary objectives, as we have seen: fecundity, healthiness of the larvae, quantity of the cocoon and silk production, improving the quality of the cocoon, and we also find disease resistant organisms. So as we can see here, the eggs, which are of different colors. We can see brown eggs and then pink, white. So based upon the breeds, these are the different kinds of colors of the eggs. So a commercial sericulture requires a good quality of silkworm eggs. So fecundity is the egg laying capacity of the breed, and it is very important factor. Then healthiness of the larvae. Healthy larvae leads to healthy cocoon crop. So healthiness is dependent on factors such as better pupation rate, less number of dead larvae uh, in uh, in this process, and shorter larval duration. So when we mean a shorter larval duration, so shorter larval duration leads to healthier cocoon, and shorter larval duration implies less chances of infection. For this reason, health of the larvae is a prime requirement for the healthy cocoon, and utmost care uh, is taken at this stage. And quantity of cocoon and silk. Quantity of cocoon produced is directly related to the pupation rate and larval weight. Healthier the larvae, more will be the pupation and the cocoon weight. So we all are aware that the formation of cocoon is based upon the larvae, and the quantity of cocoon produced is directly related to the pupation rate. That means the number of pupae, being converted, or the number of caterpillar being uh, transited into the cocoon. So if the larva is bulky, it will result in a heavy cocoon, which would obviously give rise to more number of silk. So the quantity of cocoon is also always required. So for this quantity of cocoon, the importance is given to the proper feed, which is mulberry leaves, which are adequately fed to the caterpillars. As a result, it would form into a healthy cocoon, which is quite bulkier, and it would result in a long silk. So the quality of cocoon and silk, it depends upon the number of factors, including the genetic factors. So in order to get a good quality of cocoon and the silk, we are taking a good breed of silkworm and mating with the silkworm, the where the characteristic of the hybrid is having better quality of silk and better quality of the pupae and cocoon. So quality of the silk always depends upon the genetic factors. As we have seen, there are a number of breeds of silkworm which are from various regions of the world. So in India, we have different kinds of silkworm breeds, and there are many silkworm breeds being produced by by the sericulturists. So these good quality of silkworms are taken for the production of a very good quality of cocoons, yielding a good quality of silk. So we also know about the commercial purposes which are there in advanced countries and which opt for specific breed development. They produce a specific breed which would result in a high production of silk filaments and a disease resistant worms, and they also aim for production of the sericin protein. So as we have seen, the disease resistant breeding, the major reason for crop issues is the pathogen infection, and efforts are work to evolve breeds which are tolerant and resistant to various pathogens. As we find number of diseases uh, also there in the worms. So disease resistant breeding is always required in this particular concept of breeding. So after understanding about inbreeding, outbreeding, types of outbreeding, which were outcrossing, crossbreeding, and interspecific breeding, we have dealt about the objectives of the sericulture production, where we have found X, quality of cocoon, quantity of cocoon, and disease resistant breeds. All these important objectives being required in the silkworm production. So, according to some of the one of the pioneer sericulturists, we find Leah, a scientist in the year 1993, had listed around five quantitative characteristics which are of interest to silkworm breeders. And of the five, the first one is the color of the egg. As you have seen, the color is semi-transparent and yellow color and larval markings. So in silkworm, we find some silkworm larvae are having some kind of markings, some kind of lines which are present in the larvae. And there are some larvae which do not have any kind of a lines. So these are some of the quantitative characteristics which are taken into consideration while breeding. Blood color, as we know, the insecta silkworm is having hemolymph where the blood is colorless. There is no hemoglobin present. So color of the blood is also a matter where they consider in terms of the breeding, like colorless blood or light yellow. These kind of variations in the colors of silkworm are also considered. And color of cocoon, as we have seen that the females are colored and the males are colorless. So these are some of the characteristics which are also used to identify the cocoons, male and female, and there are some more variations in different breeds of cocoons. So this is a valuable measure or quantitative measure uh, to consider while breeding silkworms. The cocoon size, as you have seen, some cocoons are having different kinds of sizes from peanut, the shape of the peanut or the shape of a pear. So we'll be knowing about these further. So as you see, egg color is very, very important quantitative characteristic for taking in breeding experiments, and these egg colors and the serosa color are taken into consideration. Then we have seen about the larval markings where some larvae are having are without any markings and some are plain. The third is the blood color. As you have seen, two major colors, white and yellow color required, which are taken for the breeding experiments. And we have seen the cocoon colors, which are normally white, green, yellow, with variations in different kinds of cocoons accordingly are taken according to the color required to pick up the breeding experiment. Fifth is the cocoon shape. So we find spherical, peanut shaped, and peaked cocoons from various regions of the world. Spherical cocoons are from Chinese varieties, and peanut cocoons are from Japanese, and pea cocoons are from the tropical varieties. These cocoon shape inheritance is controlled by polygenic additive gene actions. There is also a wide range of variations in size, surface, and double cocoon is semi-dominant over single. We also find the female cocoon is yellow and the male is white. This is also a very useful measure of sex determination where this can be used for production of high grade silk, and this measure or this quantitative characteristic can also be used to identify the female and the male. So as we know, in Bulgaria, in Vrdza, an important measure was taken to breed silkworms in the sericulture experiment station, which were directed to create more and more hybrids having high tolerance to adverse rearing conditions and having many desired qualities of silk. Initial population. So, one example of inbreeding is a female of breed NIG1, which is also having a sex limited, which is a sex limited for larval markings. And it is a Japanese type. It is crossed with another Japanese having larval marking, which is plain larval marking, and it is a Japanese male. So this particular species is KK. So we have seen about the hybridization of these species in inbreeding concept. An example of inbreeding where the male and female are crossed, and the female is a sex limited. We are interested to produce the pure lines of the female characteristics. So this experiment is taken, and the offspring is mated with again the male KK breed. So this is again repeated for nine generations, and the F9 generation is having more number of homozygous characteristics. And after uh, batch trading for these kind of generations, we find uh, selective rearing for three generations, and this resultant breed is IVA1, which is being produced, and this is extremely good variety of silkworm, which are further used in the breeding experiments and to produce a high quality of breed. So this is a popular experiment in breeding concept. So we also have known about line breeding and mutational breeding. And as we see, line breeding is a form of breeding where the offsprings are mated with the superior ancestor. And sometimes it is also the mating between cousins, sometimes the offsprings and the ancestor, as we find cousins marrying uh, or people marrying their uncles, maternal uncles. Similarly, here also there is breeding which are not very closely related breeds. So they are mating between the breed which are little distant but having the same ancestry. So line breeding is having many advantages compared to inbreeding, where we are protecting a specific pure line genetic characteristics in the future generations, and the offsprings is having the desired characteristics, and we find lesser inbreeding depression, as there is some sort of uh, a distance between the breeds and they're not very closely related. And we have found the mutational breeding, as this type of breeding is used in the sericultural experiments in sericulture silkworm breeding, as a mutation is the result. Mutation is the cause for the hybrids being produced. So this mutational breeding is extensively used in order to produce a hybrid, in order to produce a offspring which is having the mutations, and these are favorable mutation. These are having the desired characteristics. So one can find advantages of these knowledge of these different kinds of breedings which are often used in the breeding experiments and for the production of high and a better quality of breeds. So increase in the efficiency of silkworm production and requires employment of scientific methods of breeding. So we have seen in Bulgaria many breeding experiments have been taken place, and in addition we also find number of being breeds being produced in India, and these are primarily to increase the production of silkworm, as this production helps in a better and a high yielding variety of silk. So there are number of causes, a number of aspects required where we are helping for a proper cycle of silkworm, where we are taking care from the eggs larvae to the cocoon, so that all these stages of silkworm are able to pass to get a very good quality of cocoon. And utmost taking care uh, with respect to the breeding, with respect to the feeding of the larvae, with respect to the maintenance of this kind of a culture can always lead to a healthy and better quality of silkworm. So, which are the prime concern? The silkworm yielding good variety of silk is the prime concern. As we have seen, a silkworm primary the cocoon has around 800 meters to 1200 meters of silk. So this variation is seen according to the breed. There are breeds which produce around 500 to 800 meters of silk, whereas there are breeds which produce around 1200 meters of silk. So there is obvious reasons that the quantity of silk production is primary concern for the silkworm breeding. So these kind of breeding experiments, these kind of breeding are required by the breeders to produce the silk which is having a high standard, high quality, more uh, thick silk, production of more thick silk. So these are the important varieties of silk. As you have seen, all the factors contributing to the life cycle and production of a good quality of silk. Further, we'll be knowing about the different kinds of volatinism. We have seen volatinism is a concept which involves the number of breeds per year, the number of generations per year. So we'll be knowing more about this in the next classes. So volatinism, as we have seen, number of breeds per year. So there are some silkworm breeds which produces around 4 to 5 breeds per year or 4 to 5 generations per year. And they are bivolatinism, that means which produces 2 generations per year. And there is univoltine breeds which produce only 1 generation per year. So we have seen univoltine, bivoltine, and polyvoltine breeds. So univoltine breeds are often found in the northern parts of the world, like China, and these breeds are having a thicker quality of silk. And but we find that they are quite uh, resistant to diseases. They are not resistant to diseases. When we find bivoltine, they have the production is 2 times in a year and they are tolerant to some extent. Whereas polyvoltine breeds have a low quality of silk, that means the silk fiber is very thin, but it is having a high quality of disease resistant breeds. So, these polyvoltine breeds are often found in South India. So efforts by breeders are always being done to breed the high voltine where production is more in the northern parts, the breeds which are present in the northern parts of the world, to the breeds which often have many generations in a year. So these kind of mating between these breeds having a better quality of silk with the production in the year, with the number of productions in a year. So these kind of matings are often done by the breeders between the silkworm breeds to produce more quantity of silk and more the breeds which are resistant to diseases. So, so by this, I would like to conclude and having known about inbreeding, outbreeding, then in the outbreeding we have found outcrossing, then we have found crossbreeding, interspecific breeding. We have also seen line breeding and mutational breeding. Having known about all these kinds of breeding and knowing about the objectives of breeding, In addition, we have seen about some of the pioneers who have taken many aspects of the many characteristics into consideration about the silkworm breeding and about the silkworm breeding experimental stations in different parts of the world like Bulgaria and South India. So these are of primary importance in silkworm breeding. So after this class, we'll be knowing more aspects about volatinism, types of different races of silkworms in the next class. So there are different races. We find Japanese race, we find Chinese race, we find Indian race. So like this, there are number of races which are equally important, and these are valuable characteristics through which number of experiments are done for better quality of silk. So we'll be knowing about these in tomorrow's class. So by this, I conclude. Thank you.